HIV is the virus that causes AIDS. White blood cells, as shown here, are the targets of HIV. HIV it's a virus, it enters the cell, and in the cell it proliferates and makes new copies of itself. However, these copies, as they replicate, the genetic material makes mistakes during its replication, so that each virus has a slightly different DNA sequence or RNA sequence. And now these viruses are all a little bit different. Some of the viruses exit the cell, and then they can enter a new cell where they also proliferate, making similar but different viruses. The way that we treat HIV today is we use drugs called antiretroviral drugs. As shown here in the bottom right, we take our infected cells, and now when we introduce this drug, the drugs move and can enter the white blood cells as well as other cells. And now these drugs block the actions of the molecular steps that HIV uses to take over the cell. So what happens is that most of the viruses get eliminated. In fact, you can barely detect levels of the virus when patients are under therapy with the antiretroviral drugs. However, notice in these cells that one virus remained, the little red virus. Sometimes the viruses have a mutation in their DNA, and only this red one did, none of the others did. And this mutation enables the virus to survive in the presence of this drug. So over time, this virus can again propagate, and now you have an active infection where all of the viruses have this mutation. They may have additional mutations as well. These viruses, as before, can exit the cell and infect new cells, and now these new cells, which are exposed to the drug also, the virus can propagate in those cells. And these viruses are said to be drug resistant to that particular antiretroviral drug. This presents a problem for treating HIV patients in the future. There are somewhere between 30 and 40 million people in the world infected with HIV, and now there's a percentage of these viruses that are resistant to one or more of the antiretroviral drugs. This resistance can be transmitted. Thus, if a person that has HIV infects another person, they will be transmitting a strain that is resistant to that drug. Today, there are approximately 27 drugs that are approved by the Food and Drug Administration to be used to fight AIDS. These are often used in combinations with each other to try to minimize this problem of resistance. However, since we are accumulating strains that are resistant to one or more drugs, we still need to be involved in development of new drugs to treat HIV or to find approaches to eliminate it. One thing that we've noticed is in the design of new drugs, you can't consider the resistance because the drugs are new. And so what happens is that the known information about resistance to all of these drugs is really not used in the drug development pipeline. Thus, we produce new drugs that are just as prone to resistance as the old drugs. To help scientists and pharmaceutical companies address this problem, our lab has built a tool called HIVToolbox. And HIVToolbox was originally built to address the problem of integrating sequence, structure, and function. All kind of words not commonly used with a layperson, but let me just explain them in terms of your cell phone. The structure of the cell phone is, the case, what it looks like, where the microphone is, where the buttons are, where the touch screen is. Those all comprise the structure. The other aspect of your cell phone is the function. We know that the touch screen is used to enter data or select things. Your microphone is to receive audio voice. You have some buttons on the side to control volume. Those are all functions. And the sequence is the computer code in the phone that allows it to do all of the functions it needs to do. Well, it turns out that in molecular systems, they have the same points of interest: sequence, structure, and function. And what HIV does is it integrates these all into a common interactive view that helps scientists better understand how HIV works, and now in our new version of HIVToolbox to help them use existing information in the design of new drugs. So if we look at these 6 panels here, these are showing the structure of one of the HIV proteins. HIV has a small set of proteins it uses to make itself and to take over the cell. And what's shown in the little ball and stick figure is a drug bound to this HIV structure. So in panel B, the colored regions are the functional regions. For instance, the blue and brown regions are involved in making this protein bind to itself and to form what's called a dimer, and that allows it to be active. The little blue residue in the middle is a residue that's like a little molecular scissor, and it cuts other proteins normally. But in this case, the drug is binding and blocking that from working. In panel C, this is kind of a way to compare sequence. If we had 50,000 viruses, approximately, which we have in this figure, and we look at how often each amino acid in the protein changes, the ones that are colored gold didn't change in more than 99% of the 50,000 viruses. So they're super conserved. And this is information you want to consider when you're designing drugs. Now, in our new version, we have added panels D, E, and F. Now, in panel D, what we show are known drug-resistant mutations to this drug. And they have different color schemes based on what those mutations do. A mutation in red indicates that if this mutation exists in the HIV strain floating around in those cells, that it will be resistant to that drug. Okay, so you can see there's a few places where you get red mutations. A green mutation will override the red mutations, and those are called beneficial mutations. I won't go into the other colors, but one can see that there are certain atoms in the drug that are directly contacting the mutated residues. And this is the type of information that can be used to design the next generation of HIV antiretroviral therapies. In panel E, it shows how close all of the colored residues are to the drug, because the residues that are close to the drug are the ones of interest and those particular mutations. And in panel F, what's colored are the immune epitopes, or an epitope that's seen by the immune system to help it try to fight HIV infection. Now what we can do is, this is just for one drug bound to HIV, but if we take a collection of all of the drugs that bind to this particular protein, which are shown here, you can see that the resistance profiles change. And all of this information can be used in combination to try to alter the chemical drugs and their configurations to try to produce new drugs that still bind to this protein, but don't induce mutations when clinically used in practice. And that's the goal of what HIVToolbox 2 does. This project's been funded by a grant from the National Institutes of Health, and it was built by the Schiller Lab at UNLV, University of Nevada, Las Vegas. There's been 2 published papers thus far in HIVToolbox, and web links to the project page to the web application and to video tutorials on how to use it are provided. If you're interested in helping to fund the future of this project, please contact Martin Schiller at unlv.edu. Thank you.